All right. Hello to our friends joining us on the recording. Today we're going to wrap up lesson number 2. Before we wrap up the rest of this information, I want to take a moment to review with you one of the most important ideas from what we talked about in class on Monday. And this is the idea of our cells as a salty banana. So I told you we're going to be drawing these bananas like way more than you want to, right? We're going to do a salty banana cell here. So remember that I told you that calling our cells a salty banana helps us to remember where I have more of stuff. So is there more of something on the outside or more of something on the inside? That's what the salty banana tells me. So let's start with the inside of the salty banana cell. When I'm talking about the inside of our cells, what was the ion or what was the charged thing that we had a lot of inside our cells? What was there a lot inside? Yeah, exactly. When we're talking about what there's a lot of inside, our chemical abbreviation for it is K plus, or its name is potassium. Inside your salty banana cells, all the cells in your body, we have a bunch of potassium. Just like inside a banana, there's a bunch of potassium. But remember that when it's a salty banana cell, um, we sprinkled salt on the outside of that cell. So when we talk about what we find outside our cells, it's the chemical formula for salt. There's 2 ions that make up salt. Do we remember what the names of those or the abbreviations of those ions are? What do we have a lot of outside the cell? Yeah, seeing several of us chiming in. The first thing I've got a lot of is sodium. So the chemical abbreviation for sodium ion is Na plus sodium. And the other part of salt is chloride, Cl minus. Remember that the ways we figured out where these things were more concentrated was from that bar graph that we looked at together. So we had really big bars when we were looking at how much sodium we had in our extracellular fluids. We had really big bars for chloride with those extracellular fluids. But remember that when we were looking at the intracellular fluid, that was the only one that had potassium. I know I'm making you like firestorm chat me a whole bunch today. I'm going to ask you one more question. When we were looking at that chart, they labeled it intracellular fluid. The one last question I have for you, what's the other name for that fluid? What's the name that Dr. Aulis is usually going to use for that fluid inside our cells? Yeah, exactly. So what I'm usually going to call intracellular fluid, cytosol. Cytosol. Remember that that is different from what you called it in high school. In high school, you probably called it cytoplasm. Cytoplasm means that soupy stuff, the cytosol, and all of the organelles that are mixed in with it. So in our class, we're going to call that fluid cytosol. Cytosol has a whole lot of potassium in it. That's its ion, that there's a lot in it. There were 2 other things, though, that we said our cells have a lot of inside of them. One of those things is a cell's favorite food. What did we learn about last class that was our cell's favorite food? What do they love to eat? Because it's easy to eat. I mean, we all love it too, right? Yeah, exactly. Our cells do love them some sugar. The specific sugar they like is what's called glucose. Glucose is really easy to break down and make energy from. So our cells love to eat it, and they also love to hoard it. Gimme, gimme, gimme. We've got lots of glucose inside our cells. And then the other thing that we've got a bunch of inside our cells are those things that give the cells their shape or help them to do chemical reactions or do big picture functions like transporting oxygen. Yeah, a couple of us are chiming in. The one other thing that we've got a bunch of in cytosol are those groups of things called proteins. Yeah, and Lisa asked about enzymes. Enzymes are an example or a type of protein. Yeah, another one, like Asmita mentioned there, is hemoglobin. Um, so hemoglobin helps our red blood cells to transport oxygen throughout the body. Hemoglobin attaches to oxygen, and then as my red blood cells that are full of it go different places in the body, they can share that oxygen. Enzymes are what we use to break down glucose. They're what we use, we'll talk about in the next unit, what we use to read our genetic information to build proteins. So proteins as a big picture class do a lot of different things because there's so many different things they need to do. I've got a lot of them inside my cells. So quick review for us of a lot of the ideas we talked about in class last time. Our cells are salty bananas. We've got sodium and chloride on the outside. We've got potassium on the inside, along with sugar and proteins inside and outside our cell. So if we're not looking at the way that Dr. Allis drew it, if we're looking at something that looks a little bit more scientific, it's not a banana. This is kind of the general appearance of a cell with those ions on the inside and the outside. Remember from your group work at the end of our class on Monday that I asked you about, I gave you a sentence that was wrong about how a cell has a different charge inside and outside. Do we happen to remember what do I call a cell that has a different charge inside versus outside? What would I say that that cell is? Exactly. Yep. When I say that a cell is polarized, I mean that the charge that's inside that cell is different from the charge that's outside that cell. And because of that, my cell is going to be able to do different functions. So that group activity, the last question asked about a polarized cell having different amounts of water inside and outside. It polarized doesn't have anything to do with water. It's all about charge. And the way that I can have a different charge inside versus outside is because my ions are concentrated differently inside and outside. So as we start talking about what I call the tonic words, keep that in mind. Things in your body are not concentrated the same. We compare those fluids to each other to help us know where there's more of stuff or where there's less of stuff. Can anyone help me out in the chat? Which page of your notes outline do we find this terminology? Stuff about hypertonic, hypotonic. What page are we at on our notes outline? Okay, looks like we're on page 7. So for your reference, flip over to page 7. We're going to start talking about the concentrations of fluids that we have in the body. Whenever we look at fluids in the body, we're never just looking at one by itself, because knowing about one fluid by itself is not helpful. We're always going to be comparing that fluid to something. So typically in our class, there's kind of two main things that we'll compare stuff to. Sometimes we'll compare stuff to plasma, which is that fluid that's outside of red blood cells, but inside blood vessels. Other times we're going to compare stuff to the cytosol. Remember, that's the fluid that's inside the cells. So when we're talking about using these tonic words, I'm saying here's one solution that I'm going to put a red blood cell in, and I'm going to compare the cytosol of the red blood cell to the solution. That's where these comparison words come from. So if I have one solution that I would say is hypertonic to the other, a hypertonic solution has more solutes in it, or it has more dissolved things than the other solution. So when we were looking at our cell on the previous page, we would say that sodium, Na+, that's an example of a solute, something that's floating around in liquid. I can compare the amount of salt that's outside the cell to the amount that's inside the cell. The extracellular fluid compared to cytosol, we would say that the extracellular fluid is hypertonic to the cytosol because there's much more sodium outside than there is inside. A hypertonic solution has a higher concentration of solutes. There's more dissolved things in it compared to the other. Hypotonic is the opposite of hypertonic. So the hypotonic solution has fewer solutes or less solutes inside of it. So whatever has a lower concentration, we would call that the hypotonic solution. So again, if we're comparing our salt concentrations, we would say that the cytosol is hypotonic to extracellular fluid because there's more sodium outside the cell, which means what's inside doesn't have as much of it. In your body, what makes your cells happiest and healthiest is an isotonic solution. And when we talk about an isotonic solution, this is something that has the same number of solutes. The same number of solutes. When we're in an isotonic solution, the amount of something inside and outside our cell is the same, which means that we don't have to move water, we don't have to move dissolved things, we are the same. Your body likes isotonic solutions. They're good for your body. To help us understand this idea of hypertonic, hypotonic, isotonic, we're going to do a very Southern thing here, and we're going to talk about tea. So full disclosure, Dr. Ollis didn't originally come from Texas. I didn't even originally come from Oklahoma. So I'm not a tea drinker. But how many of us here in class drink tea? How many of us are sweet tea or unsweet tea fans? Let's see it in the chat. Do you like your tea? I'm curious. Sweet tea addict, love it. Unsweet tea. Yep. Yeah, we got lots of interesting. We got quite a few unsweet tea fans. Interesting. Usually the sweet tea wins it out every time. Yeah, Diana, I'm in the same boat. She's like, nope. I like it, Brianda. That would be you. Yep. It's not Dr. Pepper, says Kaylee. I agree. Whenever I go to like a formal function and there's like the water cup and then there's the cup of tea, but I'm like, but like I would prefer like a Pepsi or a Coke, but it's just like sweet tea, and that's sad to me because I'm not, I'm not a tea person. So love it. Lots of us do the tea thing. Yep, Laura, I'm with you too. I do the coffee thing. Absolutely. So when we talk about these two types of tea, we don't have the same concentration of sugar in them, right? That's what makes them different from each other. Not a trick question, I promise. Is it sweet tea or unsweetened tea that has more sugar? Who's got more sugar? Not a trick question, I promise. Exactly. Yep. So when we're comparing sweet tea to unsweetened tea, we would say that sweet tea, because it definitely has more sugar in it, that's why it's sweet. Sweet tea is hypertonic to unsweetened tea. So we're starting to use these tonic words to describe our two types of tea. The reason these tonic words matter, you've got this statement in your notes. Can anyone tell me in the chat, what page do we have this on? Are we still on 7 or have we moved to 8 now? The goal of diffusion and osmosis, we're still on 7, says Dusty. Okay, I mentioned wanting to know what page this is on because I need you to underline, highlight, star this page right here, or this statement right here. Underline, highlight, star this statement, the goal of diffusion and osmosis is to create isotonic solutions. I'm hoping you had a chance to review our packet before class today. In case you didn't, there's going to be some new words in here for us that we're going to talk about today. So one word we've already talked about is this one right here. We've already talked about isotonic. Can you remind me in the chat, if two solutions are isotonic, what does that mean about their concentration? If something is isotonic, what's up with its concentration? Exactly. Yep. When two solutions are isotonic, they have the same amount of dissolved things. Their concentrations are the same. So in chemistry, in the body in general, we've got a couple of processes called diffusion and osmosis. These two processes have a goal, or what they're trying to get to is solutions everywhere in your body that are equal. So what's going on outside the cell should be equal to what's going on inside the cell, at least according to these processes. So to help us understand these processes, isotonic solutions, we need to talk a little bit about diffusion and osmosis. All right, Dr. Allis is purposely going to crack a corny joke here. We'll see if you all like it or not. On your screen right now, you can see the original YouTube. Ta-da! Since we're not in class together, I can't tell if we actually cracked up at all. I usually get like maybe a sympathy chuckle at that. So these are the original, yeah, there we go. I got some wah-wah, yeah. These are the original YouTubes. Scientists were way ahead of their time. We had the coolest things that we could watch on YouTube before the internet existed. And by coolest things, I'm talking about we're looking at science experiments. So in these experiments here, we are looking at, I like it, Noah. No. We are looking at try to get that to go away. We're looking at a tube that has two different solutions in it. So we have on this tube, there's two sides for us to put stuff inside of it. And then in the middle, there's a little dividing membrane that keeps these solutions separate from each other. So I've got a membrane in the middle. I pour stuff in on either side, and then I just let it sit and I wait to see what's going to happen. So here's example number 2. We're doing this exact same experiment. This time we used a slightly different membrane. So we'll get slightly different results. So let's look at our solutions here. When I look at them, you have the liquid, probably water, and then we see these little red things. These little red dots that you see are things called solutes. And that's that word that you saw when we were on the first slide that we started with. Solutes are the things dissolved in water. So I can count the number of solutes or just kind of generally look at them to get an idea about their concentration. So not a trick question. Let me label our sides here for us. Does it look like side number 1 or side number 2 has more solutes? Which side looks like it might have more solutes? More dissolved things. Yeah, exactly. Not a trick question. All these little red dots that we see all over the place in this tube, these are the solutes. So side number 2 has more solutes than side number 1, which means we can start using those tonic words to compare side number 2 to side number 1. If this is the side that has more solutes, would I say that it is hypotonic, hypertonic, or isotonic, since it has more solutes? Yeah, exactly. This side has more solutes, which means that this solution is hypertonic to the other. Remember in lab last week how we talked about directional terms, and we said you have to pay attention to the order that body structures are in. So for example, if I'm comparing my head to my chest, I could say that the head is superior to the chest. It's above the chest. But if I'm making that comparison the other way, if I'm saying the chest is blank to the head, now I have to use the word inferior, right? Oop. Make sure we are muted. I'm getting a little bit of feedback here.
Do what the fuck?
Perfect. Okay. So just like we had with those directional terms, where the opposite can be used, when I say that tube number 2 has a solution that's hypertonic, I could also say that tube number 1 has a solution that's hypotonic. The concentration is lower. So the big idea here is I have two solutions that are not the same concentration. One is hypertonic, one is hypotonic. But remember from our previous slide, we said that the goal of diffusion and osmosis is to make things isotonic. It's to make them have the same concentration. These two solutions, what happens if I let that tube sit forever? I mean, I could sit there and watch it, right? Like we binge watch YouTube. I could binge watch this YouTube too, but it wouldn't be nearly as exciting as watching actual cat videos, you know. We would end up with isotonic solutions, things that have the same concentration. Now, we have the same solutions over here, so we still have the hypotonic solution on this side, and we have the hypertonic solution on this side, and we still ended up with isotonic solutions in this other tube down here. It looks different, but the way that I can say this is isotonic is based on the amount of spaced out those solutes are. So there are two ways that I can get my solutes to be as equally spaced out as possible. The body wants us to have the same amount of stuff inside and outside. Two ways to do that. First way to do that is what we see up here. What moves in my top image are my solutes. The little red dots that we saw over on this side here, they said, whoa, I am way too close to my neighbors. I'm going to sneak next door and there'll be way more space over there. So my solutes, my dissolved things, moved from one side to the other, and that allowed us to end up with solutes that all have more space between them. The concentration, one way to do it besides counting solutes is to just look at how close together or far apart things are. By the time we get to the end of this process called diffusion, everything is equally spaced out. So in diffusion, everything, the solutes, the dissolved things, will move away from each other to get equally spaced out. The way I like to kind of compare this analogy is if we were all sitting in class and I told you as you're walking into the classroom, hey, we all got to sit in the first 3 rows of our classroom. We all got to be there. We're going to be really squeezed and really close together. But about 30 seconds into class, I'd realize, oh, that's not going to work. Okay, we can fill up the entire classroom. And all of a sudden, we're all spreading out, right? We're going from a high concentration of students, of solutes. I'm spreading you out in the entire classroom. You, as the solute, moved. That's like diffusion. When our things that make the concentration, the salt, the sugar, whatever it is, that's what moves, the solutes. The other way, though, that we can end up with an isotonic solution when things are different at the beginning is using a process called osmosis. And when we talk about osmosis, we're not talking about the dissolved things moving anymore. Now we're talking about moving the we call it the solvent. That's my technical term for it. But I'll be fully honest with you, in the body, the solvent, the solution that we dissolve things in, is always water. So in osmosis, water moves. And let's look at our pictures here to see that. We started with equal amounts of water on both sides. We can see in our picture here. But I've got a lot more of the solutes, a lot more of the dissolved things in my hypertonic solution, and a lot fewer of them over here in the hypotonic solution. Well, my water molecules that are living over here on this side of my tube, they're like, I don't bump into solutes, or I don't bump into those little red dots nearly often enough, nearly as often as my water droplet friends on the other side of the membrane. So the water molecules that are on this side say, you know what? I'm going to go next door where I can find more solutes. So water moves from where there's a lot of water in solution number 1 into where it looks like there's less water, solution number 2, because there's so many dissolved things in solution number 2. So when we do osmosis, water goes from the hypotonic solution, where there's a lot of water, not as many dissolved things, into the hypertonic solution, where there's a lot of dissolved things, but not as much water. I want to pause for a moment and give us a chance to process, to ask any questions that we have. Let me check out my chat. Yeah, so Osmita asked a very important question. In the process of osmosis, do our solutes also move? Do the red dots move? And the answer to that question is no. The only thing that moves in osmosis is water, or the only thing that moves in our technical chemistry words is the solvent. The reason why that happens, just to give you a little extra information, if you look at this membrane here, the little yellow line, see how there's only teeny tiny spots or teeny tiny holes in it? Whereas over here, we've got a massive hole. When we're talking about diffusion, we need to have a bigger space for those solutes to be able to move through. If there's not enough space, only the water can move back and forth. So the water is going to go try to find more solutes to bump into. It gets tired of bumping into other water molecules. It wants to be next to those solutes. We're awfully quiet. Send me some emojis. How we feeling? Danae asked a good question as well. Danae asked, so these two things are examples of passive transport. Passive transport means it doesn't take energy, it just happens. So the question is, does this happen randomly? How do we know if we're moving water or solutes? So the first question about happening randomly, yes. Passive transport always happens because this is just what happens naturally. We'll talk about in lesson number 4, passive transport is like a rock rolling down a hill. It just happens. Whether we do diffusion or osmosis will depend mainly on the plasma membrane of a cell, honestly, in our class. The plasma membrane of a cell, there are some things that it's really good at letting straight through. Those things we would do diffusion with. But when we're talking about stuff like salt, for example, sodium does not go straight through the membrane. So in that case, if we mess with the salt concentrations, we're going to move water instead of moving the salt itself because the membrane doesn't like the salt. So that was a very long answer to say what helps us decide which one we're doing is the plasma membrane. and we're going to talk all about the chemistry of the plasma membrane next week. So great tie-in. All right, lots of smiley faces and thumbs up. So I like that. Let's take what we've just talked about here with these random YouTubes, and let's do our own YouTube experiment here. Our YouTube experiment here ties into what we mentioned before, sweet tea and unsweetened tea. So we could do a chemistry experiment together in class, and we would take that YouTube, we'd put a membrane in the middle of it, and we would pour sweet tea on one side. So we got our sweet tea over here, and we could pour unsweetened tea over here. We already talked about this a little bit, but let's remind ourselves here. When we're talking about the difference between sweet tea and unsweetened tea, it's the amount of sugar that we have in the tea, right? That's what makes them different from each other, is how much sugar they have. So let's work together here. I'm going to ask you all to help me out. Since I'm not a tea drinker, I need you to help me. I'm going to start drawing the amount of sugar over here in our sweet tea. When we get to enough sugar, tell me that in the chat, okay? Let's see how much sugar we think we need in our sweet tea. So here comes some sugar. Like I said, you tell me when we're done. We're loving our sugar. Sugar. Keep it going. Yep, keep it going. Like, you know what? Most semesters, I end up like this. Sugar, right? That's sweet tea. A couple of us are like, whoa, slow down. That's too much. One of my students one semester was like, hey, you're doing like the Olive Garden thing, right? Where they come over with the Parmesan cheese grater and they're like, tell me when you're done. Tell me when you're done. It's like, oh no, keep the cheese coming. Cheese coming. So we got lots of sugar in our sweet tea, right? That's kind of the big idea. Sweet tea, lots of sugar. Excellent. For my friends, there were a lot of us actually that said we drank unsweetened tea. Do you typically do your unsweetened tea with no sugar or just a little bit of sugar? I'm curious because I get different answers from different people. Wow, Monica's like, give it to me straight. Wow. Okay, well, if Dr. Ollis was to ever try unsweetened tea, I'd probably at least put a little bit of sugar. We're going to give it 1 sugar. I know all of my friends are like, if my unsweet tea drinking friends are like, absolutely none. Dr. Ollis isn't quite that good. I guess some of us are mentioning Splenda. Okay, that works. So we got something to make it sweeter. It's just not all that sugar that we added on the other side, right? All right, so we have done our experiment. We've set up our experiment with sugar in our sweetened tea and very little sugar. Maybe we'll say it's the natural sugar in our unsweetened tea. So we've already set this before. Let's make sure that we review it here. When I'm talking about this statement over here on the left, sweet tea is blank to unsweetened tea. We're doing a tonic word here. Is it hypotonic, hypertonic, or isotonic? Sweet tea. Exactly. Sweet tea is hypertonic. We can see it in our picture, 100%. There is way more sugar in our sweet tea than there is in the unsweetened tea. So if I flip my statement the other way around, if I'm starting with the unsweetened tea is blank to sweet tea. What would we say now if we're starting with the unsweetened tea? Exactly. Now we've flipped. Unsweetened tea is hypotonic to sweetened tea. See how we got those words, right? We were looking at the concentration of sugar. So the side that has more sugar, sweetens tea. That's my hypertonic solution. The side with less sugar is the hypotonic solution, and that's the unsweetened tea. Notice that below these statements, then, we have 2 questions to consider. We're questioning which way sugar would move and which way water would move. Essentially, we're talking about what would happen if diffusion happened. What would happen if osmosis happened. So if we set this experiment up and we tracked the way that sugar moved, or we tracked the way that water moved, how would these things move? So let's start with our sweet tea over here. We said that sweet tea is hypertonic to unsweetened tea. That means it's got a lot of sugar in it, right? Is our sugar going to want to go into the sweetened tea or out of the sweetened tea? Based on our concentrations. Exactly. Yep. Things are, I probably should have clarified this a little bit better for us. When we're talking about diffusion, the way that we can think about it is we're always going from where there's a lot of something to where there's a little of something. We always go from high concentration to low concentration. So if I have a lot of sugar in sweet tea, that sugar is going to leave the sweet tea and it's going to go into the unsweetened tea. So if we go over here to our unsweetened tea side, we kind of already answered this question, right? Sugar is going to go into that unsweetened tea. So it's going to go from the side where there's a lot of it, that sweet tea, into the side where there's not as much of it, the unsweetened tea. Hey, remind me, when we talk about the sugar moving, is that an example of diffusion or osmosis? When the sugar is the thing that's moving, the solute, shall we say? Exactly. Yep. When we talk about sugar moving, the solute moving, that's diffusion. Diffusion is when my dissolved thing moves. But remember that I might not just move the sugar. It would be possible for me to move water as well. When we talk about the way that water moves, you can consider that water is going from a place where it always bumps into other water molecules, or where I have a lot of water, into where I don't have as much water. So into the side where when water molecules are bouncing around in the tube, they're more likely to bump into a solute, a dissolved thing. So when we're looking at our tea tube here, which side or which kind of tea, let me change my question. Which kind of tea has more water in it? Which one looks like it has more water in it? What do we think? Exactly. Over here on my unsweetened tea side, because we just have that one little naturally occurring sugar over here, when water molecules are bouncing around all over the place in here, when they're moving, they keep bumping into other water molecules. And they're like, this is boring. I want to meet some solutes. So water molecules are going to leave the unsweetened tea and they're going to go next door into the sweetened tea, because that sweetened tea has all the sugar. Once water molecules pass through our membrane here, they're going to bump into sugar molecules all the time. They're going to help to dilute the concentration of sugar over there. There's going to be more water in there. Now it's going to look like there's more water, looks like there's fewer sugar molecules. We started with the hypertonic sweet tea and the hypotonic unsweetened tea. If we let diffusion and osmosis do their work, if we let the sugar move, or if we let the water move, we would end up with some really nasty isotonic tea that neither my sweet tea drinkers nor my unsweetened tea drinkers would want, right? So here's the big idea with solutions. If we put them side by side, they're always going to try to be equal. Two ways to make them equal. Either move the dissolved stuff, which in the case of our example was the sugar, or move the liquid, which in the body is always going to be water. Sweet tea and unsweetened tea, an example of hypertonic and hypotonic solutions. Yeah, so Kaylee asked an important clarifying question. Is it just the membrane that decides which happens? That is correct. So depending on whether in this tube, whether I had a membrane that had holes big enough for sugar, or whether I had a membrane that only had holes big enough for water, that would decide which of these happens. Either of them could be possible though, depending on what membrane I put in there. And when we're talking about things in the human body, we will talk about, I can't remember if it's next week or the week after, we're going to talk about how if you are something that plays nice with the plasma membrane of the cell, the plasma membrane of the cell will let you go in and out. It'll let you do diffusion. But if you don't play nice with the plasma membrane, you're not going to be able to move across it without help. So if we need to get equal, we're going to have to move the water across. Yeah, we're making some great connections to the plasma membrane in our cells. Any other thoughts about our little tea activity here? Is anyone actually drinking tea this morning? I got my coffee. Anybody got their sweet tea? Not yet. Yeah, it's a little early, right, for sweet tea. Oh, I got at least one friend though. Yep. It's never too early, right? I don't know. I'm all about coffee in the morning. Um, I do enjoy I can't remember if were you all my Coke class, right? Where we call it Coke. I do enjoy a Coke at lunchtime, but I cannot do that in the morning. Like that's just, there are lines. There are a few lines. It helps that I like coffee though, so it makes my mornings better. Yeah, maybe for lunch on the tea. Absolutely. Yeah. All right. So let's take what we just talked about in our little YouTube experiment, and we're now going to turn it into mattering for anatomy class. So the way this matters for anatomy class is because this is what happens in your body. So in your body, we have these red blood cells. Remember that red blood cells are full of hemoglobin. That was one of those proteins. It helps them to take oxygen throughout your body. Your red blood cells should look approximately like this. Let me get out my pointer here. Your red blood cells should look really flat. They're skinny, they're circular shaped. Red blood cells in your body, we should have an equal amount of water going into our red blood cells as is going out of our red blood cells. This is what happens when we're looking at an isotonic, I'll write it here, an isotonic solution. In isotonic solutions, water or osmosis is happening at the same rate of things going in as things going out. Remind me, because it's been a minute since we talked about this, when we talk about a solution that's isotonic, what did isotonic mean about the concentrations again? What does isotonic mean? Exactly. Yep. When we're talking about isotonic, we're talking about things that are equal or they have the same concentration. Think back to those teas, those two kinds of teas. When I let the amount of sugar be equal on the two sides, I don't need to do diffusion anymore. And if the amount of sugar is equal on the two sides, then the amount of water, it would be equal as well. Water goes in, and when it gets inside, it's like, oh, there's just as much water inside as there is outside, and it goes right back out. So when we have our cells in an isotonic solution, which like Seth mentioned, is 0.9% sodium. The amount of sodium inside our cell and the amount of sodium outside our cell is the same. So our red blood cells stay the right size, water moves in, and then it goes right back out. Your cells are happy and healthy when your plasma is isotonic. You start getting into trouble when we put our red blood cells in a solution that is hypotonic. Hypotonic. Remind me, because I forgot, a hypotonic solution has a lower or a higher concentration. Hypotonic. Did that mean lower or higher? Yeah, exactly. A hypotonic solution has a lower concentration of solutes. So in this case, we're talking about a lower amount of salt, a hypotonic solution. If I have a lower amount of salt, it looks like I have a higher amount of water, higher H2O. Hypotonic solutions have fewer of those dissolved things, the salt, and more of the water. And that matters because when I put a red blood cell into a hypotonic solution, the water's like, oh my gosh, there's so much of us out here. We're going in. And we're going in and we're going in and in and in because there's so much more solute, so many more dissolved things inside the cell than there is outside the cell. So what ends up happening when we put a red blood cell in a hypotonic solution is so much water goes into them, the cells bloat, they get really big like we see with these cells here. And when it gets to the extreme, your cells can actually burst. This, by the way, is the basis of the application article you're reading this week. It's called Drinking Yourself to Death, or Strange but True. Drinking too much water can kill. If you overhydrate, if you add too much water when you're drinking, you can actually cause that extra water to go into your cells. Some of your cells are cool with it. They've got space to expand with this extra water. Some of your cells are not cool with it. They don't have space to expand. So keeping the proper water balance in your body is important because if I am overhydrated, if I have too much water, that water is going to try to go into all my cells and it might cause those cells to explode. So in a hypotonic solution, water rushes into your cells, causes them to expand, and possibly explode. Yeah, I have a question in the chat. Seth asked about a 0.45% sodium chloride. Yeah, so let's put a note in here. That's an example of a hypotonic solution, 0.45%, because inside the cell, remember our number is 0.9. If we give a patient 0.45% sodium in their IV line, if we're giving them this kind of hydration, we have to, yes, like Seth said, we have to be very careful because if I give them too much of this, I'm going to end up making their red blood cells explode. Or even worse, as you're going to read about, I could end up making their neurons explode, the cells inside their brain. So we do use 0.45% sodium chloride in medicine. If someone is severely dehydrated, we can use something that is hypotonic. But we do have to use it carefully to make sure we don't end up with this. We don't want the cells exploding. That's a bad plan. So yeah, great clarifying question. We got to be careful with hypotonic solutions. We also, oops, wrong way. We also have to be careful with hypertonic solutions. So my last picture that I'm seeing here is what happens in a hypertonic solution. And remind me with hypertonic, what's going on with my concentration? Higher, lower, the same. Hypertonic. What's going on with the concentration? Exactly. In a hypertonic solution, I have a higher concentration of those dissolved things. So what that means is there's a bunch of salt out here. And the water inside my red blood cell says, oh, I'm not bumping into nearly enough salt. There's so much salt outside the cell. Peace out. I'm going to go find it. Gonna go find it. Water leaves and leaves and leaves the cell because remember, the goal of osmosis was to make inside the cell the same as outside the cell. So what ends up happening when we're trying to dilute the solution outside of us is that our cells get all shrivelly. They get all weird looking. The anatomy of our cells changes. Hey, not a trick question. If the anatomy of my red blood cells has changed, can they still do their normal physiology? What do we think? If their anatomy has changed, can they still do their normal physiology? Exactly. Yep. So many of us are chiming to the chat. No, they are not going to be nearly as effective at doing their job because now they're all shriveled up. They're in the wrong shape. It's going to be harder for them to get throughout the body. It's the same deal when we were talking about the hypotonic solutions. My anatomy was messed up over here. And if my anatomy is messed up, like you all told me, my physiology is going to be messed up as well. So it's really important that the solutions in our body are isotonic. That's why we need to stay the proper amount of hydrated. It keeps our red blood cells nice and circular, nice and flat, so they can travel throughout the body and give us oxygen. Yeah, so like Seth said in the chat, so don't drink ocean water, right? And don't put pure water right into your bloodstream. That's probably a bad idea as well. With an IV, we don't want to do pure water. So here's our application of these words here. Hypertonic, hypotonic, isotonic, and the impact it has on the cells in your body. What other questions have we thought of while we're talking about these things? Yeah, so Asmita asked a good question. Asked what percentage would we say is a hypertonic solution? A hypertonic solution is anything that is above 0.9. So it can be 1%, or when you're working in your notes packet, yeah, in your notes packet, it talks about a solution that is 3.5%. But really, that term applies to anything that is higher than 0.9. So like I said, we can do 1%, we can do 1.5, we could be really scary and say 25%, anything that's bigger. It's just a quick math thing. Anything that's higher than 0.9 is always hypertonic. Anything that's lower than 0.9 is always hypotonic. So the example in your notes outline is 0.45, but even 0.8 would be considered hypotonic, excuse me. 0% pure water would also be considered hypotonic. So it's all just referring to that isotonic number. Higher or lower than that, that's where we get these things. So Danae asked what causes sodium to go up or down in the body. It's basically what comes into your body. So what you're drinking, what you're eating, how much you're sweating. As you sweat, you lose water. So that can dehydrate you. Then it seems like the amount of salt in your body has gone up because the amount of water has gone down. So really anything that changes your water balance in your body can impact sodium. This, by the way, is why it's really good that our kidneys work so well. You'll learn about them in AMP2. Your kidneys are really good at getting rid of salt if needed, keeping salt if needed. They're really good at maintaining that balance. Any other last minute questions? I am going to try to get our room set up because we're going to start in groups here in just a moment. Let's see. All right, well, let me show you what we're going, oh, one more idea that I want to mention for you, and then we're going to split into those groups. This, I believe, is on the very last page of your notes outline, or very close to the last page. This is an underline highlight star idea for you. The idea is that water follows salt. Water follows salt. This is, if you wanted the easiest description of what osmosis is, water follows salt. So the side that had more salt in it over here, that's where the water is going to go, because there's more salt on this side. The water wants to go next door. So if you're trying to remember how osmosis works, track that water, it's going with the salt. Diffusion, the salt just moves from where there's a lot to where there's a little. So when we're talking about the way that water moves, water is going from where there's a lot of water, which is a hypotonic solution, to where there's a lot of salt, which is a hypertonic solution. For today's group work activity, we're going to practice hypotonic, hypertonic, the way that water would move, the way that solutes would move. So I've given you a couple of pictures, just random pictures of two different solutions. These solutions have different concentrations in them. And I'm going to ask you questions like, which one is hypertonic? Which one is hypotonic? You can see those concentrations. We're comparing in both of these activities, we're comparing them to each other, and we're comparing them to cytosol. So I didn't tell you what kind of solutions these are. You can make this up. You can decide, oh, this is salt, or this is sugar, or you can, I don't care what you decide it is. This is potassium. It doesn't matter. What the solution is doesn't matter as much as the numbers. So we're going to compare, the solutions to each other. We're going to compare the solutions to the cytosol of our red blood cells. And the last thing you'll do in this activity is tell me which way is the water moving, which way is the solvent or solute moving, excuse me. That is all the content that I have for you. So I'm going to pause the recording and I'll split us up into groups and give you a chance to ask any last minute questions before we disappear into groups.